All right. Now, today we're going to be talking about entomotoxicology. If you remember way back to the very, very first few lectures I gave you, beginning of this long semester, I talked about medical criminal entomology and how one of the things that we can glean from this type of entomology is the presence of drugs in a system. Okay, so this is what we're going to talk about during this lecture, is that idea that you are what you eat. So, you are what you eat. Since insects are feeding on a body after death, or parasites are feeding on a living animal, or whatever chemicals are in the body are going to be taken in by the insect. That just makes perfect sense. Over the past few decades, there has been an apparent increase in the incidence of drug-related death. Decedents in such cases may not be discovered for a substantial period of time. Therefore, the accuracy of analyzing insects in deaths involving narcotic intoxication has been subject to debate in recent years. There are only a few studies available that have explored the effect of drugs contained in decomposing tissues on fly colonization and in ovipositional behavior, or on the rates of development of carrion frequenting insects feeding on such resources. I'm sorry, there's a mosquito in here that is just buzzing around me, so if I lose my train of thought, she's trying to eat me. You may hear a smack in a minute. Stupid insect. Anyhow. So, there's relatively few studies have also examined the effects of other tissue contaminants, such as toxins or environmental pollutants, antibiotics. I told you about the body farm and how different bodies will attract different insects if they're on chemo when they died or not. So all of those different things have not been looked at, or the developmental patterns of these insects colonizing the tissues. So there's all manner of questions that are left open. How does what is in those tissues affect the attraction and the growth of insects feeding on those tissues? Now, recently, there's been an interest on the potential use of carrion-frequenting insects as an alternative to toxicological specimens in situations where traditional toxicological sources, such as blood or urine, solid tissues, are unavailable or not suitable for analysis. So the use of maggots for this purpose is well documented and has been used to assess both the cause and the manner of death. This has even been extended to analyzing insect remnants encountered with mummified or skeletonized remains to determine if maybe that body had any drugs or chemicals in its system. So, the detection of drugs. Drugs or toxins that are ingested by carrion-feeding insects are concentrated throughout the insect tissue. This includes the cuticle of the insect, in cast larval skins, in pupal casings, and in feces. All of these tissues are available for toxicological analysis. In general, the arthropods or their arthropod parts are homogenized and processed in the same way that other tissues are homogenized and processed for toxicology analysis. In the case of pupal casings or exoskeletons, a more vigorous extraction technique, originally developed for analyzing the toxicology of hair and nails, is used to ensure adequate extraction of chemicals. Once the tissues are prepped, they're analyzed. So I'm not going to go through exactly how to do the prepping of these sort of things, but there's kits you can buy to prep these things. And they're analyzed using radioimmunoassay, gas chromatography, GCMS, and thin layer chromatography. So the radioimmunoassay, this is the use of radioactively labeled antibodies to measure the amount of some substance in tissue. The antibodies will specifically bind to whatever substance we want to know about. So these antibodies are added to a liquid formulation of tissue with an unknown concentration of our substance of interest, and the number of antibodies that bind up to that substance and the number that don't can be read with a Geiger counter. So it's pretty straightforward. Antibodies will bind with their antigen. That antigen happens to be that substance we're looking for. We can read it with a Geiger counter. So this gives us the ability to infer how much of a particular substance is in the tissue sample. Excuse me. Okay, so we can get an idea of the quantity of a substance in tissue. Now, this is one of the least expensive methods used, but it is labor intensive. It takes special training and certification, what with using these whole radioactive things. And radioactive materials in labs tend to be frowned upon nowadays. People just don't want to have to deal with all of that stuff. But it is relatively inexpensive. Okay, thin layer chromatography, or TLC, is a technique used to separate mixtures. In its simplest form, it's performed on a sheet of glass, plastic, or aluminum, which is coated with a thin layer of absorbent material, usually a type of silica gel, or maybe aluminum oxide, or cellulose, anything like that. So that thin layer, hence the name thin layer chromatography. So you have this solid something covered in a thin layer of, say, silica gel. Sample is then applied to the base of the plate. So here's some example of plates. The sample is applied down here at the base. And it is placed, the bottom edge of this plate is placed in a solvent of some sort. So submerged, the bottom quarter inch or however much is submerged in some sort of solvent. When I do this in class for my students, we use acetone or alcohol. Water is even a great solvent. Any of those things can be used. So the solvent is sucked up into the plate, drawn up into the plate and through the sample. And it draws that sample along with it as it sort of wicks its way up this plate. Okay? So it sort of draws through the sample and it draws everything with it. While it's drawing this sample along with it, the different compounds in that sample will separate out. Because those different compounds travel at different rates due to the difference in their attraction to the plate, the difference in their solubility in their solvent, so their chemical makeup, all of the atoms that are involved, that causes them to travel at a different speed or different rate along this plate. Therefore, as it travels further and further, these different substances are separated out. So you can see here, I've got these 3 different compounds from this single substance that are separated out. All right. So the compounds that are separated may be colorless, but they can be stained, as you can see here. That's what happened here. They stained it with various things. And they can be then photographed using various techniques. The travel distance in TLC and thin layer chromatography is a physical characteristic of compounds. This is something that we've studied over and over again, and it's a way for these compounds to be identified. Okay, so that's thin layer chromatography. And this is something that's pretty simple to do at home with just simple paper. Use some inks. Black ink is the best. Do a dot of black ink and sort of place the very base of this piece of paper in acetone. Acetone will be drawn up, and you can see this black ink kind of smear all over the page as the different compounds separate out, and you'll see all these different colors coming out of it. It's pretty neat. All right. Okay. Gas chromatography is used to separate compounds that can be vaporized. So instead of using a liquid solvent like TLC, so like acetone in thin layer chromatography, gas is used, usually something like helium or nitrogen, something like that. The substance is moved along a microscopic layer of a liquid or a polymer, something like that, inside a glass or metal tube called a column. So the column is right here. It's a glass or metal tube that inside is coated with a liquid or a polymer. So the gas is moved along there, and the compounds being analyzed. So the compounds in this vaporized unknown, say, it reacts with those walls of the column. And the different compounds are extracted at different times. So that's one of their properties, is when they are extracted, or when they react with these different compounds. So the gas chromatograph computer, luckily it's all hooked up to computers, compares the extraction times with the extraction times of known substances, thereby allowing for identification of unknown things. So we've done this a lot. Gas chromatography is pretty common now. We have databases of different substances, when they react with that thin layer of liquid or the polymer inside the column, we know how they're going to react. We know what that looks like. When we put an unknown in there, the computer will compare and contrast that for us. All right. So GCMS stands for gas chromatography mass spectrometry. So it's basically we took that gas chromatograph and added it to a mass spec. This is awesome. So this is a method that simply combines two pretty awesome techniques. So the GC is the same as we just saw, same as normal. It uses gas, it separates out substances from an unknown sample and helps identify them. Then it goes into, so it takes out those unknown substances bits at a time, separates them out, and then sends them into the mass spec. What the mass spec does is it captures and ionizes those separated compounds and determines some of their chemical rather than physical properties. So when they separate out of a full compound, that's a physical property, right? So now we're going to add on a little bit more. We want to know a bit about their chemical properties. So using the information from these two systems together, a computer can positively identify really, really complex compounds. So this is right now the gold standard for forensic toxicology equipment. Whenever you see somebody saying they ran a toxicological analysis on some blood and they found heroin and aspirin and these other things, this is what they're doing. All of those compounds were separated out into their very base bits. They were analyzed, the amounts were analyzed, and the computer kicked back what that would then be. Neat. All right. The advantage of using maggots instead of, say, bodily tissues for toxicology assessment can be seen in chromatography. So, chromatographs obtained from larval extracts present less contamination than those obtained from human body samples. This is especially true if the body samples are decomposed. So, it's like the maggots will take in these chemicals and then keep them from degrading, keep them from being contaminated by other weird things, from products, from microbes, from whatever. So it's like a neat storage system for these chemicals. Hooray. Toxins in tissues are transferred through the food web, basically. And what happens during this transfer from things that are eating other things is they are bioaccumulating. So basically you get maggots that are feeding on tissues. Those maggots will uptake the chemicals, then beetles will come along and feed on the maggots. And so you get this high amount of toxins in beetles that predate on blowfly larvae. So this bioaccumulation, it's the accumulation of substances, such as pesticides or drugs or something like that, that builds up in an organism. So bioaccumulation, the accumulation of substances that builds up in an organism. This occurs when an organism absorbs a toxic substance at a rate greater than at which that substance is lost. So, we are able to excrete certain substances. We can get rid of them, we can pee it out, we can sweat it out, whatever. If an organism is unable to do that, unable to get rid of it, it just stores it somewhere and it bioaccumulates. Okay, so this bioaccumulation is especially useful in environmental studies. A study in the 80s showed that it was possible to detect a mercury in larvae, pupae, and adults reared on mercury-contaminated fish. So we were able to see mercury bioaccumulate up throughout these trophic levels or throughout this food web. An interesting case study along these same lines saw an entomologist use similar procedures where the body of a woman was found in an advanced state of decay. They were unable to do any toxicological analysis. She was covered in maggots. She was in a rural region of an island, and there were distinctly low mercury levels present in the maggots, which indicated a probable geographic origin of the victim, an area almost free of mercury contamination. So that was really neat. It gave an idea of place of origin of this person based on this mercury contamination, or in this case, the lack of mercury in these maggots. So bioaccumulation is most potent in maggots. They're feeding directly on tissues and in those predators feeding on the maggots. So the higher up the trophic level it gets, the better off we are. The substances will carry through life stages, but each molt and metamorphosis will get rid of some of those chemicals. So we'll see if these maggots only were, say, exposed to these chemicals in their first instar, we'll still see residual of these chemicals in the third instar or in the pupal stages, but it'll get less and less. So maggots may have very, very high concentrations of a toxin, which will lessen during the pupal stage and lessen further in the adult stage. Adult flies are still useful for toxicological analysis, but they may have a much lower amount of toxin. Also, bioaccumulation means that toxin is concentrated. Therefore, we can't use or at least at the moment, we can't use the amount of toxin in a maggot to determine how much toxin was present in the dead body. It builds up, it changes from life stage to life stage. So right now we're unable to analyze that. We can only give a yes or no answer. Was there this toxin in this body or not? So we use it a lot of, was there heroin in this system? Or is this particular drug in this person's system? Because if it was there, then the maggots had it. All right. So impact. The ingestion of toxins can affect the growth and development of insects. Various toxins have been studied. So let's look at a few that have been published about. Cocaine, for example. Maggots that are reared on tissues contaminated with cocaine show differing developmental times based on the amount of cocaine present in the tissues. If the tissues have sublethal dosages of cocaine, so not enough to kill a person or an animal, maggots will develop at approximately the same rates as maggots that were not exposed to the drug. So there's no real difference, at least no significant difference in between developmental times. If the tissues have lethal doses of cocaine, however, so really high levels of cocaine, so in the case of a body that died of cocaine overdose, the maggots develop much more quickly than those not exposed. So the maggots look bigger, they look older, and the developmental time or time of colonization may be off. The pupal stage, however, is the same, and mortality rates are not affected. So the maggots don't die from a cocaine OD. Interesting. They just grow really, really fast. Morphine. Morphine results in more rapid development and the production of larger maggots regardless of the dosage. So even a little bit of morphine can cause a much more rapid development and larger maggots. So once again, an overestimation of the time of colonization. The pupal period is also larger or longer. So it appears to be proportional to the amount of drug present at the tissues. So the more drug, the longer the pupal stage. So there's an error of up to 29 hours. So over a day of error rate is possible if these maggots are used to determine postmortem interval. So you can be a full 24 hours off if you're doing this time of colonization estimation. The same patterns were observed in maggots using heroin. Maggots using heroin? Awesome. I had an awesome picture in my head of a maggot just shooting up heroin. Anyhow, of maggots ingesting heroin. They had faster developmental time. They produced larger maggots until a maximum maggot size was attained. This also caused an overestimation of the postmortem interval. So that's a problem. So when maggots were exposed to methamphetamine, the larvae seemed to develop very quickly, but the pupal mortality was very, very high. They just could not make it through that pupal stage. So those that did make it through the pupal stage often failed to have viable offspring. So this had a negative impact on the life of these insects. Amitriptyline, which is a common antidepressant and also used in low dosages for pain management, long-term pain management, showed no significant effects on developmental rate, but it did cause substantially longer post-feeding and pupal stages, and the larval mortality was very high. So that wandering stage of the maggots and the pupal stages were much longer than normal, and a lot of the maggots died. In blind studies, attempting to calculate PMI, or time of colonization, using maggots fed on drug-contaminated tissues. It was demonstrated just across the board that there was a substantial underestimation of the postmortem interval due to the general trend of maggots speeding up their developmental response due to drugs. So there's a big problem with not knowing if there are drugs in the system. They also looked at pesticides. So common pesticides, things like parathion, malathion, things that are commonly used on cropland. These can cause intoxication of humans and animals by either inappropriate or deliberate use. This pesticide is detectable in various arthropod species of Diptera, of Coleoptera, Hymenoptera, Isopoda, Acari, so ticks, which were collected during all decomposition phases of animals contaminated with the pesticide. So the pesticide caused the arthropods to be abnormally small. So different from some of these other drugs. It also presented a deterrent to colonization at normal colonization sites, such as the mouth. However, it didn't cause alteration on insect succession. So this parathion, this caused insects to not normally or not colonize a body in a normal manner, especially if that insecticide was drunk, was eaten in some way. So it caused a reaction in those insects. They just did not lay their eggs in the normal places. Malathion, on the other hand, caused significant delay in insect oviposition and a significant change in the species of insects colonizing the remains. So you saw different species that were able to colonize. So in a case where you have insects on a body that you're not expecting, you expected an entirely different type. So let's say you didn't get a lot of caliphorids on that body and you expected them to be there. You may have pesticides on that body. That would be something to look into. Okay, now the collection protocol. If you are to use insects in toxicological analysis, you must collect them in a particular way in order to preserve the evidence. All collection equipment must be clean and, if possible, washed in hot water. You don't want to have any type of contamination, cross-contamination with outside chemicals. Tissue, fluid, and insects must be collected in vials free of preservatives or any external contamination. Insects can be collected just in the normal way with forceps, spoons, paintbrushes, whatever you want, and nets. In most cases, a refrigerated storage before the analysis is recommended, and they should be sampled in individual glass vials, labeled with proper information, taken to the laboratory where larvae are killed by freezing rather than boiling. This will help to preserve that toxin. For the collection sites, samples can be collected on the corpse where insects are commonly found, in the clothes or around the corpse. It doesn't really matter where you collect them, just as long as they're associated with that body. And when you analyze these, many samples for analysis are best sent in their original state. However, others require additives to maintain them in optimum conditions until they reach the lab. Part of the samples should be stored in individual vials and refrigerated as reference samples. The freeze-killed larvae should be rinsed in distilled water, while the pupa should be rinsed with methanol prior to extraction to avoid contaminants. The sample can then be prepared for analysis, and this preparation depends on the substances to be analyzed. So we won't go over those individual protocols. If you are sending in samples to be analyzed to a toxicology unit or something, it is best to ask the toxicologist what he or she prefers, if they just prefer you to send in the freeze-killed insects and they'll take it from there, or if they prefer to have you add something to those insects. With that, if there are any questions, you just let me know.